Welcome to the Deep Dive. This week, well, it's the usual story, isn't it? The longevity biotech field is just constantly churning out new research.
It really is. Sometimes it feels like trying to, drink from a fire hose, just keeping track of it all.
Exactly. So you sent over the latest list from longevitypapers.com, and that's what we're tackling today, the very newest stuff relevant for researchers like you. Think of it as maybe a curated shortcut.
Yeah, a way to sift through the noise.
Mm-hmm.
And let's be upfront, right? In a field like this, most weeks aren't about huge, earth-shattering breakthroughs.
No, definitely not.
It's more about incremental progress. And this week, honestly, falls mostly into that camp. Interesting work, yes, but steps forward rather than giant leaps.
Right. We weren't exactly expecting the Fountain of Youth paper this week, but we were looking for those interesting little nuggets, maybe surprising findings.
From credible places, yeah, things that might just sort of nudge our current models or point in a slightly new direction, even if it's super early days for that idea.
Precisely. So our mission here is to pull out the key bits from these papers, help you see what's worth noting, and importantly, why, without getting totally bogged down.
And based on that, good institutions, surprising angles, potential model shifts, we've kind of zeroed in on 3 papers from this week's list.
Okay, great. Let's jump straight into the first one then. It's called Boosting Cellular Longevity Through Intracellular ATP Modulation
Right.
by Oz, Sue, Patnaik, and others at the University of Virginia School of Medicine. It's a preprint, just hit Bioresorf, that's pronounced bioRxiv, on April 16th. The abstract basically says, aging is damage accumulation, ATP levels drop with age, but maybe supplementing ATP could help, at least in yeast. So targeting energy metabolism. Let's unpack that.
Well, what's interesting is how direct they're being. They're not just tweaking a signaling pathway. They're actually trying to directly change the ATP levels inside the yeast cells. And their main finding really is showing this link. Keeping intracellular ATP steady seems to extend yeast lifespan, which, fits with the whole energy metabolism and aging story.
So it reinforces that link.
It does. But the direct manipulation is the notable part here. Makes you wonder, if this could ever translate, could tackling energy supply so directly be a powerful approach? Big if, of course.
And the novelty, like we said, maybe incremental. We know energy is important, but this specific method.
Exactly. It builds on known concepts, but the how is interesting, and there were definitely some surprising bits.
Like what? What stood out?
Well, first off, the way they got ATP into the yeast, they used a nucleotide transporter called NTT1,
Okay.
which comes from a parasite. That's
Yeah.
a pretty clever bit of, biological borrowing.
Mm-hmm.
And they suggest maybe 2 ways it's working. One, the imported ATP might just reduce the cell's need to make its own energy, suppressing catabolism.
Right. Less internal effort needed.
Or 2, maybe the yeast are sensing ATP outside the cell, and that's triggering other pathways. They mentioned MAPK and mitochondrial pathways specifically.
So like an external energy available signal. Did they actually see lifespan extension?
They did. In Figure 4D, they show about a 40% lifespan extension in the yeast under their best conditions. And the stats look solid. That p-value is tiny, 4.03 times 10 to the minus 18.
Wow. Okay. So very unlikely to be chance in their model.
Right. But, and this is but Figure 6B shows that too much ATP was actually toxic.
Ah, the dose makes the poison. That classic U shape.
Exactly, hormesis. Too little isn't good, too much is bad, just right is beneficial. What was really weird, though, was that inducing mitochondrial dysfunction.
So messing up the cell's powerhouses?
Yeah, that actually rescued the cells from the ATP toxicity.
Wait, really? Making the mitochondria work less well made the cells less sensitive to too much ATP. That seems counterintuitive.
It does. It suggests a really, really delicate energy balance inside the cell. Maybe if the mitochondria aren't firing on all cylinders, the cell is somehow protected from being overwhelmed by this external ATP flood.
Complex.
Very. And they also did RNA-seq, looking at gene expression. Figures 3D and E suggested the added ATP suppressed glucose metabolism and, interestingly, autophagy.
Autophagy. Isn't that usually seen as good for longevity, the cellular cleanup crew?
Generally, yes.
No?
So suppressing it seems a bit paradoxical. If the overall outcome is lifespan extension, it definitely complicates the simple more energy is always better narrative.
Okay, yeah, that definitely makes you pause. So given all that, what are the big limitations here? Reasons to be skeptical?
Well, the number one limitation is the model organism, yeast.
Right.
It's great for basic mechanisms, but yeast aging is just fundamentally different from human aging. No stem cell decline, no immunosenescence, none of that complexity.
Got it.
Also, that 40% lifespan extension. It sounds good, But for yeast, it's actually kind of modest compared to some other interventions like calorie restriction.
Okay.
The toxicity issue is obviously a huge red flag for any potential therapeutic idea.
Yeah.
And crucially, zero mammalian data in this paper. That's a massive leap needed for translation.
Right.
Oh, and one more thing, they give p-values showing statistical significance, but no effect size metrics.
Yeah.
So it's hard to gauge the sort of practical magnitude of the lifespan change beyond just it happened.
Okay. So interesting mechanics in a simple system, but a long, long way to go. What experiments would you want to see next?
Oh, definitely need to see if this holds up in mammalian models. Cell cultures, maybe iPSCs, eventually mice.
Yeah.
And identifying those putative ATP sensors in yeast and seeing if humans even have analogous pathways, that's critical.
What about combining it with other things?
Yeah, combinatorial studies would be fascinating. ATP modulation plus, say, rapamycin. What happens then? And maybe looking at ATP's role in specific age-related disease models, not just overall lifespan.
Okay, let's move to paper number 2. This one's titled HDAC11 Deficiency Regulates Age-Related Muscle Decline in Sarcopenia. Authors Renato Odria and others from a group in Badalona, Spain. Published on PubMed April 12th. The abstract basically says knocking out HDAC11 in aged mice helped with muscle atrophy, mitochondrial function, and fat burning in the muscle.
Right. And sarcopenia, that age-related muscle loss, is a huge deal for healthy aging.
Absolutely. So this sounds pretty relevant.
It does. What's compelling is it tackles a major aging issue, sarcopenia, directly in mice, a mammal. Finding that inhibiting HDAC11 seems to, well, maybe even reverse some aspects of muscle decline in old mice is significant.
And HDAC11 is potentially druggable.
Yeah, that's the other key piece. There are apparently selective inhibitors being developed or available, which makes you think more immediately about potential therapies down the line.
Okay. And the novelty again was described as incremental, building on other HDAC work but focusing on this specific one, HDAC11 in older animals.
Pretty much. HDACs are a known area, but HDAC11 is less studied, especially in aging muscle. But they certainly found some surprising things.
Such as?
Well, one thing that jumped out was the mortality data. They reported almost no deaths in the HDAC11 deficient aged mice before 22 months, unlike the normal mice.
Wow.
But got to temper that small sample size for that part of the study. So, need caution there. Could be survival bias.
Okay, noted. What else?
They showed pretty clearly, figures 2b to d, that the age-related shrinkage of muscle fibers was reduced. The deficient mice had fiber sizes closer to young mice.
That's quite striking.
Yeah. And improved structure at the neuromuscular junction, the nerve-muscle connection, figure 3D. Also, metabolically, they saw better fat burning in the mitochondria, fewer fat droplets in the muscle, figure 6AB.
Okay.
And they even saw reversal of some age-related changes in the types of fatty acids in the muscle, improving the omega-6 to omega-3 ratio, figure 6E.
Interesting. Anything else?
One more key thing, satellite cells.
No.
Those are the muscle stem cells needed for repair.
Right.
They found more satellite cells in the HDAC11 deficient mice, figure 7A, and also showed these mice were better at regenerating muscle after an injury, figure 7AD.
Okay, that's quite a list of benefits in the muscle system. But you mentioned the mortality sample size. Any other caveats or limitations?
Definitely. Besides the small mortality cohort, they didn't track muscle mass changes over time longitudinally. It was more of a snapshot.
Okay.
And seeing better structure at the neuromuscular junction doesn't automatically mean better function. Need to test strength and nerve signaling directly.
Good point.
And a big one, diet wasn't controlled. That could definitely confound the results about fat metabolism. Were the mice eating differently? We don't know.
Right, that could muddy the water significantly. So what follow-up experiments are needed here?
Well, first, redo that mortality study with way more animals and proper postmortems to see why they lived longer, if they did.
Makes sense.
Using inducible knockouts where you can turn off HDAC11 just in the muscle at a specific time would be cleaner.
Isolate the effect.
Exactly. Need longitudinal imaging to track muscle mass over time. Definitely need in vivo functional tests of those neuromuscular junctions electrophysiology.
Test the actual signaling.
And diet control studies are essential, maybe even looking at the gut microbiome alongside the fat metabolism stuff.
Okay.
Delving deeper into those satellite cells with culture work and single-cell RNA-seq would be great too. And figuring out what proteins HDAC11 actually targets in muscle that requires proteomics.
And testing actual drugs.
Absolutely. The crucial next step is taking those selective HDAC11 inhibitors and testing them in normal aged mice to see if you can replicate the knockout effects pharmacologically.
Got it. Okay, final paper for the deep dive this week. Dietary methionine restriction started late in life promotes healthy aging in a sex-specific manner. This is from Ululu Hernandez Arcega and team at the University of Pittsburgh's Aging Institute. PubMed, April 14th. Now, methionine restriction isn't new news in longevity, right?
Not at all. It's a well-established intervention in model organisms. But the key here is started late in life.
Right. That's the potentially translational part.
Exactly. Because realistically, getting humans to restrict methionine from a young age is unlikely. But if it works when it started later, that's much more relevant.
So what did they find in the mice?
They reported improvements in quite a few areas in aged mice, started late on the diet, better neuromuscular function, better metabolic health, even better lung function, and just generally less frailty.
Okay. Any particularly surprising findings in this one?
Well, just the breadth of improvement seen even with that late start was encouraging. And interestingly, they also saw benefits in a mouse model of Alzheimer's disease. And it didn't cause weight loss, which can be a concern with dietary restriction.
That's good.
And they did some neat single-nucleus RNA and ATAC sequencing in muscle. Found that different cell types within the muscle actually respond differently to the methionine restriction. It's not a uniform effect.
Highlighting that cellular complexity again. But there's a fairly significant but with methionine restriction, especially regarding human data, isn't there?
Yes, absolutely. This is crucial context. While the mouse data often looks promising, a human trial, albeit relatively short, I think 8 weeks didn't show a significant effect on epigenetic clocks.
And those clocks are considered pretty important markers of biological age, right?
They are. So that discrepancy is really important. Plus, the benefits in this mouse study were noted as being sex-specific. That needs a lot more investigation to understand why and what it means for humans.
Okay, so late life start looks promising in mice, potentially relevant, but that human epigenetic clock data and the sex specificity are major caveats. What's next for this line of research?
Definitely need replication in other mammals, not just mice. Trying to figure out the optimal dose and duration for methionine restriction in older humans is key if this is ever going to be practical. And really digging into why there are sex-specific effects is critical for understanding translatability.
Okay. And now those were the 3 we focused on, but the list you looked at from longevitypapers.com had, well, a lot more papers this week.
Oh yeah, a ton. It really covered the gamut.
Can you give us just a quick flavor of some of the other topics?
Sure. There was stuff on tRNAs and CNS diseases, blocking TREM1 for pulmonary fibrosis, glycation and HIV aging, ATF6 in skin senescence, circulating DNA and frailty, PFAS pollutants and biological aging, environmental enrichment for stroke recovery, inflammation, and cardiometabolic issues.
Wow.
Mitochondrial quality control in blood stem cells, the mitochondrial stress response, how using tech affects cognitive aging, brain signal variability, rare longevity gene variants in MTR signaling,
Keep going.
senescent cells in wound healing, even somatic mutations and coral aging, plus MRI brain aging in monkeys, epigenetic aging in HIV, tachykinin and lifespan, lipid balance in B cells,
Okay.
genetics of physical activity, brain gradient changes with age, ginseng and C. elegans, a new get-up test for older adults, APOE and Alzheimer's, a urolithin derivative for cognition, VDAC1 in Alzheimer's.
What was there? Deep breath.
Methyl donor nutrients, physical activity, and epigenetic clocks again. Heme and liver cell senescence, stress granules and testicular aging, extracellular vesicles and muscle aging, exercise and heart changes, BCL-LOMNA and stem cell aging, plant compounds affecting NAMPT, NTORC1, and muscle metabolism. I think that's most of them.
That is an incredibly diverse list. It really shows the breadth of the field. So it sounds like while that work is happening, the 3 we discussed maybe had those slightly more surprising elements or potential model shifting hints this week.
I think that's fair, yeah, even if they're incremental shifts. This week's papers overall, they reinforce key themes we see a lot. Mitochondria, inflammation, targeting specific pathways.
Right.
But they also really hammer home the challenge of model systems. Extrapolating from yeast or even mice to humans is always fraught with difficulty.
That constant refrain.
Absolutely. Rigorous follow-up, replication, controlling for confounders. It's just essential. No shortcuts.
So wrapping up this week's deep dive then, key takeaways. ATP modulation in yeast, interesting mechanism, foundational, but miles away from humans.
Pretty much.
HDAC11 deficiency in aged mice. Looks promising for sarcopenia, druggable target, but needs much more validation, especially on safety and the mortality finding.
Agreed. More near-term potential perhaps, but requires care.
And late-life methionine restriction shows potential relevance for starting interventions later, which is good, but that human epigenetic clock data mismatch is a serious point of caution.
Definitely warrants caution, yes.
So for everyone listening, as you dive into your own work, keep these nuances in play. No single silver bullet this week. Probably not most weeks, right?
Unlikely. It's about fitting the pieces together. Each study adds a small piece to that massive, complex puzzle of aging.
So here's a thought to leave you with. If these incremental findings, these little nudges, keep pointing towards similar core pathways, mitochondria, epigenetics, inflammation, metabolism, what might the cumulative impact look like in, say, the next 5 years? And maybe more importantly, where do you see the most fertile ground for truly game-changing discoveries in longevity biotech?
Yeah, worth pondering. And definitely worth digging into the full papers if any of these topics overlap with your interests.
Absolutely. We'll be back next time, keeping an eye out for whatever the research world throws at us.